So in biochemistry, the first thing is pyrimidine and purines synthesis. So de novo synthesis of purines, pyrimidines, and purines. In order to synthesize purines and pyrimidine, which are the nucleotides important in DNA, purine and pyrimidines, first we need glutamine. And in order to convert glutamine into the further precursors, into the further compounds, we need carbon dioxide along with 2 ATP molecules and a synthase enzyme, because we are going to build something. So we need synthase. What synthase? We need carbamyl phosphate synthase. Synthase is an enzyme. So carba from carbon dioxide, ATP give the phosphate, and we convert this protein into this amino acid into carbamyl phosphate, and at this point we add aspartate, which is another amino acid. So after adding aspartate into carbamyl phosphate, so now we have to convert this into orotic acid. So we need to dehydrogenase this compound. So we remove the hydrogen, dehydrogenase, and by removing this dehydrogenase, so now we have compound known as orotic acid, and this dehydrogenated orotic acid dihydroorate dehydrogenase enzyme, and this dehydrogenase is inhibited by leflunomide. If we inhibit this, we cannot make orotic acid in the DNA. Pyrimidine synthesis is inhibited. So this is a type of antineoplastic immunosuppressant or antimetabolite medication work. So in orotic acid, we need to add phosphate pyroribosyl, and so we need PP, which is a ribose sugar product. So we convert ribose 5-phosphate and make it a ribose sugar. So in order to make a nucleotide, we need sugar and amino acid base. So this is the orotic acid. So now we have to synthesize something. So we add a synthase enzyme. What synthase? We need UMP synthase enzyme. This synthase enzyme is impaired in orotic aciduria, which we will discuss in a while. So orotic aciduria, impairment of the synthase enzyme, cannot make the UMP. So there is orotic acid in whole the body, especially in the urine. So we have orotic aciduria. And when UMP is formed, it is readily converted into diphosphate, and this diphosphate is going to this diphosphate is now converted into cytosine triphosphate. Obviously we add a lot of phosphates and we deaminate UDP into cytosine. and this cytosine goes to the DNA replication or DNA synthesis process where it add in the nucleotide chain. Either the UTP goes to produce cytosine or the UDP goes to make thymidine. So in order to make thymidine, we need to reduce this compound. So we need reductase, UDP reductase, and this UDP or nucleotide reductase, this reductase is inhibited by hydroxyurea. If we inhibit this enzyme, we cannot make thymidine, so we can stop the pyrimidine synthesis. So we add reductase to make it a diuridine diphosphate reductase. We make it deoxyuridine diphosphate. So this diphosphate is now converted into monophosphate. So this is the main precursor which is going to make thymidine. For thymidine, we have known that by methylation of the uridine or uracil we can make thymidine, because in the thymidine THYMI we have the mi methyl TH, which I will describe later on in a while. So deoxyuridine monophosphate is now methylated by an enzyme known as by a methyl donor. So we add this and it will generate deoxy monophosphate, deoxythymidine monophosphate. So we convert this after donate this thymidine. So now this after giving this methyl group, methyl donor, trihydrofolate is converted into dihydrofolate, which is reduced to make trihydrofolate, and which is then add methylene in it. So this processes again and again be used. So here we can make deoxythymidine monophosphate. Here we make cytosine triphosphate. We can also make UDP or UMP. So how to inhibit this reductase enzyme in the bacteria? We can use trimethoprim, which inhibits the bacterial tritetrahydrofolate. In human body, methotrexate inhibit dihydrofolate reductase enzyme and all causes side effect in the human body. And in the protozoa we have the pyrimethamine. It inhibits the protozoal dihydrofolate reductase enzyme, and which block the pyrimidine synthesis. So no nucleotide, no DNA precursor, so there is inhibition of DNA or anti-metabolite drugs. So for this is all the pyrimidine formation. In purine it is pretty much simple. We use ribose 5-phosphate, convert it into synthase. We add synthase. What synthase? We PRPP synthase. This PRPP is going to make inosine monophosphate, which is converted into AMP and GMP. For GMP, inosine is dehydrogenase. So this phosphoribosyl pyrophosphate is inhibited by a medication known as 6-mercaptopurine or 6-mercaptopurine or azathioprine. And this dehydrogenase enzyme, it is inhibited by this dehydrogenase. It is inhibited by mycophenolate and ribavirin. This is how these antimetabolites work and also causes side effects in human body. So this whole lecture is about the Novo synthesis of pyrimidine, how they are formed and how different enzymes interact with each other to make this pyrimidines. And the next thing is we have discussed that orotic acid urea, it means that increase orotic acid in the urine. And this orotic acid in the urine is increased because of the mutated or abnormal UMP synthase enzyme. If this is not present, there is increased level of orotic acid in the body, so no UMP is formed. So in treatment we have to bypass this enzyme and by giving UMP or uridine triacetate, treatment of orotic aciduria. Abnormal UMP is no UMP formed, so we cannot make it can lead to megaloblastic anemia. Megaloblastic anemia. Obviously, we do not have we do not have thymidine, we do not have UMP, we cannot make any of the thymidine, adenine, cytosine, or guanosine metabolites. So these metabolites or these precursors are important in DNA synthesis. So when there is no DNA synthesis, so it can lead to macrocytosis or megaloblastic anemia. And this megaloblastic is refractory type, which means that either we give folic acid or vitamin B12 cobalamin, but there is no effect or no resolution of the megaloblastic anemia. Megaloblastic anemia is still there, so it's a refractory type of megaloblastic anemia, and the patient is having failure to thrive, and it cannot failure to thrive, there is developmental delay, and also there is no hyperammonia, which is pretty much important for differential diagnose or ornithine transcarboxylase enzyme carboxylase enzyme deficiency, in which there is also orotic aciduria, but it is associated with hyperammonia. So this is pretty much important in pyrimidines and purine formation. For a nucleotide, we have the base, we have a sugar, ribose sugar. These are the general information, and we need a phosphate. So nucleotide, phosphate. Nucleotide is a phosphated sugar and ribose, whereas nucleoside is only base plus sugar, ribose sugar, either DNA or RNA. For purines, they are pure as gold, so adenine and guanine are the purines, whereas the pyrimidines, we cut the pyramid. So pyrimidines are cytosine, uracil, and thymidine. If we deaminate cytosine, we can get uracil. If we deaminate adenine or guanine, we can form xanthine and hypoxanthine. And if we deaminate also, if we methylate uracil, we can get guanine. So if we deaminate methylated cytosine, we can get methylated uracil, and methylated uracil is guanine. Also, there is a word cytosine and guanine. They have triple bond, and increase in the content of cytosine and guanine in the nucleotide chain, we have the increased melting point of the DNA chain. And what are the requirements for making the pyrimidine? In order to make pyrimidine, we need only aspartate and tetrahydrofolate. But if we need to make purines, we need aspartate, we need glycine, we need glutamine, and also we need tetrahydrofolate. So this is all about the de novo synthesis of purines and pyrimidines.